Okay, our next speaker is Mike Stubenvoll. He will tell us about ATX-2, the C. elegans. Ortholog of human Ataxin-2 regulates centrosome size and microtubule dynamics.
Good morning. I want to thank you for the opportunity given me to present our research here for you today. As mentioned, my name is Mike Stubenvoll, and I am a PhD student at Oakland University in Rochester, Michigan. And I'm going to be talking today about the RNA binding protein ATX-2 and its involvement in centrosome assembly. So the centrosome is a dynamic organelle found in animal cells. It's composed of two perpendicularly arranged tubular structures called centrioles, surrounded by a matrix of other proteins and molecules, the pericentriole material, which contains the factors necessary for proper microtubule nucleation and anchoring. The centrosome serves as the main microtubule organizing center in animal cells, and as such is necessary and a vital component of the bipolar mitotic spindle to ensure the fidelity of cell division. Now, since each dividing cell requires two centrosomes, the centrosomes themselves must duplicate once per cell cycle. And extensive work in C. elegans has outlined five core factors that are required for this duplication process. This process appears to be evolutionarily conserved throughout the animal kingdom, including in humans. Loss of any of these five factors results in centrosome duplication failure. One of these factors is the kinase ZYG1. Shown here is a ZYG1 mutant embryo that has undergone centrosome duplication failure marked by the presence of monopolar mitotic spindles. The SUSY20 gene encodes an RNA binding protein and was discovered to negatively regulate ZYG1, as loss of SUSY20 function in ZYG1 mutants restores centrosome duplication and bipolar spindle formation. However, the full molecular mechanism of this regulatory process is not yet well understood. And so to this end, we sought to identify factors that associated with SUSY20 in vivo. And using a biochemical approach, we performed SUSY20 immunoprecipitation and analyzed the co-precipitants by mass spectrometry, and we identified the RNA binding protein ATX-2 to co-precipitate with SUSY20. Now, since SUSY20 and ATX-2 are both RNA binding proteins, we were wondering if their interaction was mediated by RNA. However, performing immunoprecipitations after removing RNA still yielded ATX-2 in the co-precipitating, suggesting that their interaction is independent of RNA. We next asked if ATX-2 played a similar role to SUSY20 in the developing embryo, and we found that depletion of ATX-2 by RNAi resulted in roughly 50% embryonic lethality. Now, to understand what was causing this embryonic lethality, we immunostained the embryos and observed a number of different cell cycle defects, polar body extrusion failure, spindle alignment, spindle misalignment, and chromosome segregation defects. However, one of the most prominent defects was cytokinesis failure. This is a single-cell embryo exhibiting a tetrapolar spindle in which the centrosomes have duplicated and the DNA is replicated, but the cytoplasm has failed to divide. Further, double depletion of SUSY20 and ATX-2 resulted in enhancement of both the embryonic lethality and cytokinesis defect phenotypes, suggesting that ATX-2 and SUSY20 share a positive or exhibit a positive genetic interaction. We next asked if ATX-2 could also suppress ZYG1 as SUSY20 can, and so we depleted ATX-2 in ZYG1 mutants and indeed found that depletion of ATX-2 partially restored centrosome duplication and bipolar spindle formation. Further, double depletion of ATX-2 and SUSY20 in ZYG1 further enhanced that restoration, thereby indicating that ATX-2 does indeed negatively regulate centrosome assembly and further supporting the positive genetic interaction that we had observed. And so to understand how ATX-2 might coordinate its actions within the cell, we immunostained embryos for ATX-2 and observed a distinct cytoplasmic localization pattern that was almost completely abolished in the ATX-2 mutant. Interestingly, SUSY20 mutants appear to have less cytoplasmic ATX-2 than our wild-type controls, and indeed quantitative analysis of the cytoplasmic ATX-2 levels indicated that our ATX-2 levels in SUSY20 are roughly half that seen in wild-type. Further, ATX-2 levels appear unchanged in the zygote mutant, leading us to suggesting that ATX-2 is positively regulated by SUSY20, with both functioning upstream to negatively regulate zyg1. Now, since loss of ATX-2 can restore bipolar spindle formation to zygote 25 mutants, we asked how that might be occurring, and we reasoned that by inhibiting ATX-2, we might increase zyg1 activity. And so we measured zyg1 levels at ATX-2 mutant centrosomes and observed a roughly twofold increase in zyg1 at the ATX-2 mutant centrosomes, leading us to believe that in the zyg1 mutant, elevated zyg1 levels compensate for the reduced zyg1 function and permitting the successful centrosome duplication and bipolar spindle formation. Now, zyg1 has been shown to be a regulator of centrosome size, and so we asked what effect this increase in zyg1 had on the centrosome size in ATX-2 mutants. And so we looked at the PCM factor SPUD5 and found that, again, there was an increase in centrosomal SPUD5 levels. However, cytoplasmic SPUD5 levels remained relatively similar. We saw a similar result in when we looked at SPUD2 as well, suggesting that centrosomal PCM components, or the centrosomal levels of these PCM components are increased while the cytoplasmic levels and overall levels remain the same. So we next looked at gamma-tubulin, which is a microtubule nucleation factor under various conditions via time-lapse microscopy. And we found that in ATX-2 RNAi embryos, there was a roughly 2.5-fold increase in centrosomal gamma-tubulin levels. Interestingly, this was similar to the increase in centrosomal gamma-tubulin that we see in the SUSY20 mutant. In the ATX-2 mutant, which we have shown is a near no mutant phenotype, we see an even further increase in centrosomal gamma-tubulin levels. In addition, we see the similar thing in SUSY20 mutants, in which we perform ATX-2 RNAi. And while the centrosomal levels here are increased, quantitative Western analysis shows that overall gamma-tubulin levels within the embryo remain unchanged. And our data indicates that ATX-2 cellular levels of ATX-2 are inversely correlated with centrosomal gamma-tubulin levels, but not total gamma-tubulin levels within the embryo. And so SPUD5, SPUD2, and gamma-tubulin are all required for, are all essential components of the microtubule nucleating process within the embryo. And so we wanted to look at what effect this had on microtubule dynamics in ATX-2 mutants. And so we utilized the strain, and I apologize that the movie is not working, but if you'll appreciate these, black dots here, these time-lapse movies allowed us to track the growth of these microtubules away from the centrosome. And the first thing we noticed was that at ATX-2 mutant centrosomes, there's an increase in this GFP-EBP2 signal. And so what we did was, through chymograph analysis, we quantified the number of microtubules that grew out from the centrosome and observed a significant increase in microtubule nucleation in the ATX-2 mutant. To see what effect this had on the growth of these microtubules out towards the cortex, we performed chymograph analysis at the cortex, and we actually observed a decrease in the number of microtubules that grew out to the cortex. And this suggests that the increased microtubule nucleation seen in these ATX-2 mutants results in an overall negative effect on microtubule growth. And in C. elegans, it's been shown that microtubule growth is regulated by both a microtubule stabilizing complex, which contains the factors ZAG9 and TAC1, and also is reliant on free tubulin concentration. However, in ATX-2, TAC1 levels, both total and at the centrosome, are increased compared to wild type. So we reason that this defect in microtubule growth is likely not due to a reduction in microtubule stabilization. Therefore, we reason that increased microtubule nucleation might be leading to a depletion of free tubulin, which resulted in the stunted microtubule growth. And so we overexpressed GFP tubulin in ATX-2 mutant embryos, and we indeed found that this overexpression resulted in a reduction in the number of cytokinesis defects. These cytokinesis defects would be a result of the improper microtubule growth. This reduction in cytokinesis defects translated to a slight but significant reduction in embryonic lethality as well, suggesting that the increased microtubule nucleation was resulting in a depletion of free tubulin and therefore stunting microtubule growth. We next thought that if we increase microtubule nucleation by overexpressing a microtubule nucleation factor, in this case gamma-tubulin, we could elevate microtubule nucleation factor levels at the centrosome and further inhibit microtubule growth by enhancing microtubule nucleation. And we indeed found that to be the case. In ATX-2 mutants, where we overexpressed gamma-tubulin and centrosomal gamma-tubulin levels were increased, we observed an increase in cytokinesis defects and a very slight increase in embryonic lethality as well. Now, we reason that the increased microtubule nucleation that we see in ATX-2 was responsible for the cytokinesis failure phenotype that we see through affecting microtubule dynamics, and we thought that if we reduced centrosomal TBG1 levels, we could partially restore cytokinesis to the ATX-2 mutants. And indeed, when we performed TBG1 RNAi in the ATX-2 mutant, we saw a reduction in both the number of cytokinesis defects that we saw, as well as a reduction in embryonic lethality, further suggesting that elevated levels of microtubule nucleation factors contribute to the cytokinesis defect we see. So, to summarize, we've shown that loss of ATX-2, shown here in red, contributes to elevated levels of PCM factors and overall larger centrosomes. These larger centrosomes possess more microtubule nucleation factors, leading to excessive microtubule nucleation, which results in stunted microtubule growth and cytokinesis failure. And so to conclude, we have shown that ATX-2 is positively regulated by SUSY20, with both acting upstream of ZAG1, and ATX-2 acts upstream in this pathway to negatively regulate centrosome size and microtubule dynamics. And with that, I'd like to thank the members of our lab, the funding sources, including the GSA, for providing me with the travel award to come talk to you today, the CGC for strains, and Wormbase for all their wealth of information. Thank you.
Thanks. Does Ataxin-2 bind microtubules directly?
We have not seen that. The cytoplasmic localization pattern that we've noticed, we haven't noticed any binding of ATX-2 to tubulin, or not that I can actually remember off the top of my head. Sorry, I'm a little nervous. But we're more focused on the ATX-2 RNA interaction, and that's where our direction is headed next.
That was really nice. You shouldn't be nervous. It was great. I thought I noticed in the ZAG1 ATX double that there was a phenotype where the AB and P1 anaphases looked synchronous, and I wondered if you had other polarity phenotypes in the ATX-2 loss of function.
So,
yeah,
this one here?
Yeah.
So there is a significant cell cycle delay in the ATX-2 mutants. In this case, in the ATX-2 ZAG1 double mutant, while some cases we see the restoration of the bipolar spindle formation in relatively normal embryos looking like this, the ATX-2 phenotype is very strong, and so we do see that this is actually an embryo that has undergone cytokinesis failure. So though it is in the ZAG1 mutant background,
one cytoplasm.
This has undergone cytokinesis defect due to the highly penetrant ATX-2 phenotype.
Thank you.
Mm-hmm.
Is ATX inhibiting ZAG1 solely at the protein level, or is it possible that there's any transcriptional regulation?
Well, so that's one thing that we're interested in. We don't know at the moment if it's transcriptionally or if it's just spatial control of the protein localization, but that is something that we are interested in finding out.
Does that ATX defect translate to any human disorders?
Does it, I'm sorry, what?
Does this ATX mutation relate to any of the human disorders?
Oh, so yes, ATX-2 is the ortholog of human Ataxin-2. Ataxin-2 is implicated in neurodegenerative disease. Ataxin-2 is also an RNA binding protein. However, this is the first case that we've had any indication of the role of this particular protein in centrosome assembly and cell division.
Okay, thank you.